Okay. Good afternoon, all. Please let me know if I'm audible or not.
Yes, sir. You are audible.
Okay.
Yes, sir.
Good afternoon, everyone. I welcome you all, my dear friends, my dear students, for this guest lecture session. Today we have Dr. V.S. Adhao sir as a guest faculty. Sir will delivering guest lecture on topic different factors affecting enzyme activity. So I take this privilege to introduce Dr. V.S. Adhao sir. Dr. V.S. Adhao sir completed his post graduation from MGR Medical University, as well as PhD from Dr. Babasaheb Ambedkar University, Uttar Pradesh. Sir presently working as assistant associate professor in a department of pharmaceutical chemistry at Dr. Rajara Gode College of Pharmacy, Malkapur. Sir has near about 11 year teaching experience as well as research experience. Sir published more than 15 articles in national as well as international level, as well as sir present seminars in national as well as international platforms. Currently, sir involved in method development and validation of different antiviral drugs, as well as crystal engineering and molecular imprinted polymer techniques. Okay, so I thankful to Dr. V.S. Adhao sir for accepting our invitation and delivering the seminar or giving guest lecture on factor affecting enzyme activity. Thank you, sir.
Thank you very much, JK sir, for nice introduction here. So, shall we continue the session?
Yes, sir. Please.
Okay, thank you. So, dear students, very good noon to all of you. Today we are going to discuss about different factors that affects the activity of enzymes. So, as we have already started in previous lecture, I discussed with the Ambure madam that introduction to the enzyme was already completed here. So today we are going to study about different factors that affects the enzyme activity. I am Dr. Vaibhav Adhao. I am also working with your same institutions. So here we will start with the little bit a small introduction to the enzymes. So let me ask here, what do you know about the enzyme? Can anyone tell me? Please be interactive during the session so that the knowledge will be shared with us. So can anyone tell me or throw a light over the topic enzyme here, or a very small definition of enzyme? You can also use the chat box, and I can see the chat box. Or someone unmute yourself and tell me, what do you know about the enzymes?
Enzymes are the biological catalysts which increases the rate of reaction.
Okay, always try to give one by one answer. I will give chances to all peoples. Okay, so I request again all peoples mute all of you. Okay. So we will continue with the enzymes, as we know that these are the biocatalysts of the chemical reactions that occurs in our body. So this enzymes being a biocatalyst, the word catalyst is nothing but some species which is helpful for carrying out the reactions or speed up the chemical reactions. But the word biocatalyst that we use here, as it is acting as a substance which is being used here to carry out the chemical reactions in the living organisms. That's why the name comes here, biocatalyst. These enzymes are neither consumed or not permanently altered here themselves as a consequence of their participation in the reactions. And with the exceptions of this catalytic activity by RNA molecules, or we can say ribozymes, all enzymes are protein in nature. I am again repeating the statement. All enzymes are protein in nature, but all proteins are not the enzymes. You might heard about this thing because enzymes are proteinous in nature. Exception was there for the RNA molecules or ribozymes that are not the proteins in nature. So this enzyme actually works here. You can see the image here. This enzyme fits. झूम केलेला आहे हे दिसतंय सगळ्यांना. आता बघा इथे जेव्हा आपण बघतो इथे की एन्झाईमला एका पर्टिक्युलर साईटला इथं बाइंड करावं लागतं मॉलिक्यूल सोबत. So these active sites which were present over the enzyme, on this active site, the molecule exactly fits here. So it is having certain mechanism of action that also we have to discuss. So if it is having a proper shape, proper geometry of the molecule, which is similar to that of the enzyme space here, that can having the proper interaction between the enzyme and a substrate molecule. So that's why a very important property of enzyme was discussed here, that our enzymes are specific in nature. So specificity of enzyme is very much important. You can compare here two images. In the first images, you can see there is exact correlation, exact matching of the geometrical shapes with the enzyme and substrate. So this is the right circle, or we can see here the circle which is over U. It can be exactly fits with this molecule, and this molecule can be fit here exactly in the first case. But if you alter the orientation of this molecule substrate, it is not being fit here. These two sides are not being able to carry out here the annotation. So this annotation I can't make here. So here you can see here this purple color or pink color box, square box, cubic box, it is not being fit with the circular part, and this circle shape or spherical shape is not being fit with the cubical shapes here. So this is having a specific activity over the enzyme. Once the substrate is having a typical geometrical shapes that exactly fits with the enzyme, then and then only the enzyme substrate complex will form. Because under the mechanism of enzyme substrate complex action, we must see here how the enzyme action is being carried out. As we know that the basic enzymatic reaction can be represented here as the substrate is being added with the enzyme that gets converted into products and enzyme is regenerated. This is a very basic reaction among the enzyme. As we all of us know that the enzyme not being consumed here and not being utilized here for the carrying out reaction, but it is directly being utilized and finally it is being converted again into the its original form. But here the substrate and enzyme binds together so as to form the enzyme substrate complex first, and after forming the enzyme substrate complex, it is being converted into the desired product that we want to create here. And after the product is formed, the enzyme is liberated free to carry out a reaction with another molecule of substrate. So this enzyme is regenerated. In this reaction, the E is representing the enzyme which is catalyzing the reactions here. S is the substrate molecule, P is the product, and ES is representing as the enzyme substrate complex. Now you can see here, enzyme is a very big molecule as it is said to be protein in nature. So you can compare here. Here the glucose molecule, which we're going to bind here at this particular site of enzyme. So this green color molecule is a protein molecule which is acting as an enzyme, and this enzyme gives or provides the spaces here or active sites here to bind the glucose molecule to this enzyme. Once this glucose molecule binds to the enzyme, it forms the enzyme substrate complex. You can see here the shapes which are available in this are easily accommodating the glucose molecule, and that binds to the particular site here. So it shows such a type of a reaction molecule enzyme substrate complex here. And once this enzyme substrate complex molecule form here, it alters the substrate molecule into the product. And once it alters into the substrate molecule of this product, it can be giving us a final product. We will take one schematic representation here for the catalysis action of the enzyme in this slide. Now, the enzyme catalysis, that is being action of enzyme, is being schematically presented here with you. Let us assume here a molecule A being acting as a substrate here, that is going to be bind with the enzyme. And once it is being bind with the enzyme at its particular active site, you can see this active site here, and this active site will be considered here for giving us a action over the molecule species. So here what is happening, a molecule A that is acting as a substrate, it binds with the enzyme. So this enzyme and substrate complex were formed here. You can see a green color and red color. They are finding a complex here as an enzyme substrate complex. And this substrate is having its specific geometry to bind over the enzyme action in this. So due to this reason, it is being formed a proper geometrical shape gets attached to the enzyme molecule so as to form the enzyme substrate complex. After the formation of enzyme substrate complex, the catalysis will take place here. The reaction is going to convert the substrate into the product by utilizing the enzyme here, and this enzyme forms the enzyme product complex after the enzyme substrate complex. So enzyme substrate complex gets converted into the enzyme product complex. The red colored substrate gets converted into yellow colored. In this schematic representation, I have taken these two colors. So this yellow color generated enzyme can be reutilized here for the further binding with the molecule A. So such a type of reactions are continuously going on in our body. So this enzyme catalysis can be occur constantly in our body. Now, how this enzyme mechanism will be takes place, that we will consider. There are several different models are available for the enzyme actions. So there are two different types of models. Generally we can see the third one also, but most popular are the two enzyme substrate models. The very first one is lock and key model, and second one is induced fit model. So I am directly jumping towards the images here. So in the first image, you can see here the lock and key model. Okay, so under the lock and key model, you can see here the substrate molecule and enzyme molecules in the blue color and substrate in the green color. You can see here the enzyme is exactly having the same geometrical space where the substrate can exactly fit. In similar manner, a lock is having a specific key. At the active site, it is thought to be a rigid, inflexible shape that is being exact complement to the shape of substrate. And the substrate fits in the active site much like a key fits in the lock. So due to this reason here, this model name itself as a lock and key model. But here, this mechanism is most commonly known here for the enzyme specific action. But there are certain enzymes that can changes the shapes according to the shape of substrate. So that theory was evolved later on. So that theory is given by Koshland's induced fit model here. So this is being induced fit model. Now you can see here with these two models, you can compare the shape of enzyme in the first images. That A, it is having the exactly the similar kind of geometry that is substrate can fit here so as to form the enzyme substrate complex. But in the case B here, that is induced fit model, here the substrate is having a specific shape, but enzyme is not having a exactly similar kind of shape in it. So this enzyme is found to be flexible. So the scientist Koshland has given the induced fit model, and this active site is found to be flexible. So this another criteria of this enzyme is also evolved in it. So this enzyme is giving us proper shapes here for the substrate, and this substrate exactly fits over the enzyme molecule. So once it is being fit here by adjusting themselves towards the substrate, how the substrate will come here and this similarly binds with the enzyme, and enzyme substrate complex gets formed here. So this type of mechanism will be generated. In general, जर सोप्या भाषेत आपल्याला सांगायचं झालं तर एन्झाईम आणि सबस्ट्रेट हे एक असं कोरिलेशन आहे की जसं टीचर आणि स्टुडंट्सचं असतं. जसं तुम्ही आता फर्स्ट इयर बी फॉर्मला इथे ऍडमिशन घेतली, इथे 4 वर्ष तुम्ही शिकणार. टीचर काय करतात, इथे तुम्हाला नॉलेज देतात. तुम्हाला जर बुक्स मधनं भेटलं असतं तर टीचरची गरजच नसती राहिली, स्कूल कॉलेजेस तिथे निघालेच नसते. मग त्यामुळे आपण काय करतो, 90% जे आहे हे तुम्हाला स्टुडंटला बुक्स मधनं इथे स्टडी करत असतात. 10% रोल जो असतो तो टीचरचा असतो. तर टीचर काय करतो, इथे तुम्हाला या प्रॉपरली गाईड करतात जिथे तुम्हाला डिफिकल्टी येतात इन सिमिलर मॅनर ऍज दॅट ऑफ द एन्झाईम. एन्झाईम्स पण इथे काय करतात, प्रॉडक्टला इथे पेसअप करतात, स्पीडअप करतात आणि त्याला एका प्रॉपर स्टेबल प्रॉडक्ट मध्ये इथे कन्वर्ट करतात सबस्ट्रेटला, जेणेकरून त्याची एक प्रॉपर लेवल तिथे तयार होईल. So as to get such a type of activity in similar to that of substrate and enzyme, I am comparing here substrate as a student and enzyme as a teacher. So teacher moves the substrate towards the product. So a teacher is moving the students from a raw to the perfect individual so as to get a proper job and proper earnings to them. So here this substrate and enzyme binds together for a few time, like you peoples for 4 years you are binding with us, and after 4 years you are going for your future, and teacher will remains here in the college itself so as to teach the new batches. आता या वर्षी तुम्हाला नवीन टीचर्स इथे मिळाले, पुढच्या वर्षी आणखी नवीन टीचर्स मिळतील, म्हणजे एक एक वर्ष जे आहे स्टेप बाय स्टेप तुम्हाला इथे पुढे तिथे सरकवल्या जातं. सो टीचर प्लेज अ व्हेरी इम्पॉर्टंट रोल इन द स्टुडंट्स लाईफ so as to solve the difficulties in it. Okay, so in similar manner, enzyme is also providing the space for the substrate so as to carry out the reactions very quickly so as to achieve the desired aim of this particular substrate in it. So now this is a little bit introduction to the mechanism of action of the enzyme. Okay, now there are several factors which affects the enzyme activity. Before going to check the different factors, I would like to concentrate here one terminology associated with the enzyme reaction kinetics, that is thermodynamic perspective of the mechanism of enzyme action. As of now, we know that these all enzymes been accelerating the rate of reaction by providing the transition state with the lowered activation energy for the formation of transition state here. So you can see in the image the transition state. In each and every reaction we have the transition state, because this transition state is a very highly energetic moiety and highly unstable moiety. So substrate gets converted into product via passing through the transition state. If we are carrying out the reaction without enzyme, it requires a much more energy. You can see here the black color curve here, which is having the black color curve having the transition state, which is not having utilizing the enzyme. And if we utilize the enzyme, the transition state is being achieved earlier without utilizing the much more energy in it. So you can see here the lower activation energy for the pink color line in this case. So this lower activation energy means that of the more molecules have required the energy to reach the transition state here. So enzyme plays a very important role as such in the image. So this thermodynamic changes in this image, you can see here a very first reaction A, that reaction has to be carried out without enzyme. So a person is moving here a big stone, spherical stone here from this cleft here. He has to push this stone here, and later on it could be carried out. So here what is happening here? In this case, the molecule is being shifted towards the very high altitude here, and the activation energy without the enzyme can be studied here. So this net enzyme activation energy without enzyme, it requires more energy, and whatever the product we get here, it is having a very less net energy release by splitting from the substrate to the product. But if we carry out this reaction with the enzyme here, and this enzyme will be acting as a glucose molecule, or we can say a lactose, and the enzyme lactase we are utilizing here so as to split into the glucose and galactose here. So this glucose and galactose can be splitted here by utilizing the enzyme. So the height of this peak is being very low, and we required a very less activation energy with the enzyme. And due to this reason here, it generates the more net energy being released here, because energy is being not consumed here for converting the molecule. So this enzyme rate of chemical reaction, it is being described here by a different molecules and reactant that can be converted into the products in specified unit time. So it is being described here by the international units, that is IU, and one IU is defined as the activity of the enzyme which transforms 1 micromole of substrate into the products per minute per liter of a sample under the optimal conditions and definite temperature, and it is expressed as IU per L. It can also have another unit as known as a Cattle unit, and this is Cattle or catalytic unit we can say here. One catalytic unit is defined as the number of moles of substrate which are transformed per second per liter of a sample, and it is abbreviated as a CAT or a small a. So these are the units that we have to discuss over the enzyme kinetic action. Now we are moving towards our main content of heading, that are the factors affecting enzyme action. So there are several factors or numerous factors which affect the reaction rate of enzyme. So few of them, mostly most important factors we have taken here. One here, and whenever you have any doubt, please raise your hand and talk to me, or you can also put your question in chat box if you do not understand it. Okay, so we will move towards the very first factor, that is effect of substrate concentration, which is a very important factor among the factors affecting enzyme action. As we know that at the lower concentration of the substrate, the active sites of most of the enzyme molecules are not filled completely. So because there is not much substrate available. So due to this reason here, the higher concentration causes the collision between the molecule, and rate of reaction increases with the first order at the initial stage. So whenever we are considering the effect of substrate at its maximum velocity in it, and this reaction is reached with the active sites which is almost continuously filled. Whenever we have the increased concentration of substrate after the sub point here, it will increase the rate, and reaction rate therefore increases as the substrate concentration increase, but it offs the level. So it is said to be the zero order reaction. Now we're moving towards the one chart here. You can see a shape of curve that relates to the activity of this substrate concentration in a hyperbolic manner. So you are getting the hyperbolic curve here. Initial phase of this curve is simply representing here the molecular concentration or substrate concentration exactly matches with the enzyme concentration. So all the substrate is being correlating with the enzyme and binding with the enzyme, so as it is giving the first order reaction initially. And once all the sites over the enzyme gets completely bind, even though we increase the concentration of substrate here, it is not having the proper sites to bind for the substrate. So substrate remains as such intact. It is not forming the enzyme substrate complex. So this hyperbolic curve having the three different phases in it. At point A, you can see here at point A or B, it is increasing or decreasing the concentration of substrate. The reaction will react accordingly. So due to this reason, the enzyme substrate complex will form with the corresponding change in the initial velocity of the reaction. The rate of reaction, which is initially depending on the concentration of substrate. That's why it is said to be the first order reaction. You might have studied the thermodynamics in your 11-12 standard. So this thermodynamics, first order, zero order, second order reactions are there. So initially up to the point B, we can say here it is a first order reaction. As we increase the concentration of substrate, the rate of reaction also increases accordingly. But whenever, even though we are increasing the concentration of substrate later on, the rate of reaction is not being further increased accordingly. So here, what is happening here? As the all binding sites of enzymes remains intact here, bind with the substrate, and enzyme substrate complex get formed. There is no free site available to bind with the excess quantity of the substrate. And due to this reason here, we can say that this molecular concentration will be achieved here, and this molecular concentration will be depending upon the substrate concentration of the molecule. So at point C here, this essentially all the enzyme which is present as the enzyme substrate complex, since no free enzyme remains here as available for forming the enzyme substrate. So further increase in the concentration of substrate cannot increase the rate of reaction. So that's why we are getting the straight line here after some point which is being achieved, the maximum rate of reaction. So it remains the steady state concentration of substrate, and it is having the plateau here. So they said to have a zero order reaction for the substrate concentration. Generally, I represented the Km here on the x-axis and Vmax for the reaction. So you can see here the Vmax will be representing here as a maximum velocity of the concentration, and Km is representing the Michaelis-Menten constant here. This Michaelis-Menten constant Km is the substrate concentration at which the V1 is exactly half of the maximum velocity. So that's why it is said to be the Vmax by 2. As a point is a B point that is exactly the half velocity of the reaction. So that is a maximum velocity will be achieved here, and Km is representing the Michaelis-Menten constant. Thus, the Km has the dimension of the substrate concentration, and it is being specific and constant for the given enzyme under the defined conditions of the time and temperature and pH. So this Km can be determined here, the affinity of enzyme for the substrate, and lesser the Km of its affinity and vice versa. So due to this reason, the Km value helps in determining the true substrate for the enzyme action in it. So this kind of reaction will be occurred here for the Km and its significance in it. So here, this is a very important factor in the case of substrate concentration. Here, the Michaelis-Menten equation was also studied, but due to time constraint, I am skipping that topic because it required at least 1 hour to solve this in the Michaelis-Menten equation. So I am directly focusing here over the effect of concentration of substrate and different other factors in it. Okay, so this is a very important factor in it. Okay, so we are moving towards the next slide now. That is effect of enzyme concentration. So under the effect of enzyme concentration, you can see here as we are increasing the amount of enzyme, the rate of reaction also increases gradually. But if there are more enzyme molecules than are needed, adding additional enzyme will not increase the rate of reaction. So the reaction rate therefore increases as the enzyme concentration increases. But again, the same factor, if the substrate is finished, the enzyme remains as such. तुमच्याकडे जर जास्त एन्झाईमचं कॉन्सन्ट्रेशन असेल तरी तो पर्टिक्युलर लेवल पर्यंत इथे आपल्याला इन्क्रीज झालेला दिसून येतो आणि लॅटर ऑन जेव्हा आपल्याला सबस्ट्रेट हा संपून जातो पूर्णतः तेव्हा परत सुद्धा आपल्याला इथं हायपरबोलिक जसं की आपण सबस्ट्रेट मध्ये बघितला त्यामध्ये पाहायला मिळतो. बट ऍट द इनिशियल स्टेज इट गिव्स अस अ व्हेरी first order reaction, that is first order reaction. As the concentration of enzyme increases, the rate of reaction also increases and up to the certain limits here, and later on even though we increase the concentration of enzyme, if there is not availability of the substrate, then it can be simply said that it is having the steady state concentration over it. Okay, so this is a very important factor again. So you can see here different other factors on it. Okay, so enzyme concentration, it is also the directly proportional to the rate of reaction. So we can say that as the concentration of enzyme increases, the rate of reaction also increases. Okay, so this is a second factor that is a very important in it. Now we will move towards the next factor, that is effect of product concentration. So under the effect of product concentration, we know that the enzyme activity declines when the product starts accumulating, and this is called as product or feedback inhibition. Product inhibition, or we can say it is a feedback inhibition. एक सिंपल लॉजिक आहे याच्या मागे की जेव्हा आपण एखादा सबस्टन्स जो आहे तो स्टार्ट करत असतो, हा स्टार्ट केलेला आहे. From the start of reactions, enzyme reacts with the substrate. Some intermediate complexes were formed like enzyme substrate complex, then enzyme product complex, and finally enzyme is regenerated. So enzyme having the three different phases. You can see the image here, enzyme 1, enzyme 2, and enzyme 3. In the enzyme 1 state, this enzyme is unbound or not bounded to the substrate, and it is being converted into intermediate as enzyme substrate complex. So this enzyme substrate complex is having a enzyme 2 types of mechanism. So this enzyme 2 is having a binding of substrate over it, and enzyme 2 is being converted again into the enzyme product complex first, and once it is converted into the enzyme product complex, later on it is being converted into the product and enzyme is regenerated here. But what is happening here under the case of feedback inhibition here, the presence of product inhibits the enzyme 1, means whatever the free form of enzyme is being inhibited here due to the with the substrate, and this binding of the substrate will be affected due to the accumulation of product over the active sites of the enzyme, and due to this reason here the enzyme is not freely available to bind with the next molecule of substrate that is being converted into the product. So this type of inhibition is known as feedback inhibition, or it is also known as product inhibition. So it inhibit the activity of enzyme. हे काय करतात ना, स्टॉप करून टाकतात एन्झाईमची ऍक्टिव्हिटी, जेणेकरून जेव्हा प्रॉडक्ट बाइंड झालेला असेल एन्झाईमला तर पुढचा जो सबस्ट्रेट मॉलिक्यूलला एन्झाईमला अटॅच करायचा आहे, जसं आपण सांगितलं की एन्झाईम इज रिजनरेटेड सो एज टू बाइंड विथ द अनदर मॉलिक्यूल ऑफ सबस्ट्रेट. मग इथे काय होते, एन्झाईम रिजनरेट होतो आणि रिजनरेट झाल्यावर तो प्रॉडक्ट सोबतच बाइंड राहतो आणि त्यामुळे सबस्ट्रेटला तिथं चान्स मिळत नाही बाइंड होण्यासाठी, म्हणून तिथं एन्झाईमची ऍक्टिव्हिटी थोडी रेट ऑफ रिएक्शन हा कमी झालेला आपल्याला इथं दिसून येतो. आणि कशामुळे कमी होतो, प्रॉडक्ट जो आहे हा आपल्याला बाइंड झालेला दिसतो एन्झाईम सोबत. So this accumulation of product over the active sites of enzyme can be said to have a feedback inhibition on it. So under certain conditions here, the reverse reaction may be favored, forming the back the substrate. म्हणजे काही केस मध्ये काय होतं, मग एन्झाईम तोच वापरलेला राहतो. Some reactions are reversible in this case, and whenever the product can be converted into the substrate. So this is also known as a product inhibition reaction, or reversible reaction can be occurred at a minimum equilibrium of the reaction. So that's why we have to take this product from this product formation, and the enzyme is regenerated. So such type of a continuous chain reaction you can see in the biochemistry life cycles here, and this life cycles having the several steps in it, so that the product is immediately being altered into the another substrate by utilizing another enzyme in it. So this is a product concentration effect. As the product concentration increases, the rate of reaction also decreases gradually. Okay, याचा इन्व्हर्सली कोरिलेशन राहत, जसं प्रॉडक्टचं कॉन्सन्ट्रेशन ते वाढत जाईल, त्याचा रेट ऑफ रिएक्शन हा आहे थोडा थोडा मॅक्सिमम पॉइंट अचीव केल्यानंतर कमी कमी होत जात असतो. ओके, सो दिस इज थर्ड अँड इम्पॉर्टंट फॅक्टर. अगेन पहिला आपण फॅक्टर बघितला इफेक्ट ऑफ सबस्ट्रेट कॉन्सन्ट्रेशन, दुसरा बघितला इफेक्ट ऑफ एन्झाईम कॉन्सन्ट्रेशन, आणि तिसरा आपण बघितलाय इफेक्ट ऑफ प्रॉडक्ट कॉन्सन्ट्रेशन. मग आता हे तीन कॉन्सन्ट्रेशन जे आहे हे मेनली रिएक्शनशी कोरिलेट राहतात. Now there are several other factors like temperature, pH, time, effect of activator that also alters the rate of reaction. So we will take first factor amongst the other factors, that is effect of temperature. These are the different 7 factors that we have to study here for the effect of substrate concentration over 8. So this effect of temperature, that raising the temperature increases the kinetic energy of the molecule. As we know that whenever we are giving certain amount of heat for any kind of a reaction, the molecule gets the energy from the heat, and the kinetic energy of the molecule also increases. And due to this reason, they increases their motion. So therefore, the frequency with which they collide, that also increases. So this combination of more frequent and more highly energetic and productive collision, that increases the rate of reaction among it. So this temperature factor can also be studied. As we increases the temperature, that can be helpful here for the enzyme action. So now we say that this enzyme are the biocatalyst. Generally biocatalyst आपण म्हटलं तर ह्युमन बॉडीज मध्ये सुद्धा ह्या रिएक्शन इथे व्हायला पाहिजे. मग आता ह्युमन बॉडी मध्ये असं कुठलं टेम्परेचर रेंज आहे की ज्यामध्ये आपल्याला मॅक्सिमम हे मिळू शकतं? कोणी सांगू शकेल मला? व्हॉट इज द नॉर्मल ह्युमन बॉडी टेम्परेचर? यू कॅन टाइप युअर आन्सर्स इन द चॅट बॉक्स. माहितीये मला ही दुपारची वेळ आहे, झोपेची वेळ असेल तुमची, पण झोपू नका. यू टाइप युअर आन्सर्स इन चॅट बॉक्स. What is the normal human body temperature? Very good. Very first answer is given by Neha Nimkarde, followed by Tuja Khetri, then Sakshi Mamankar, Vaishnavi Hudekar, Ayuri Mankar. Very good. 37 degrees Celsius is a normal human body temperature. Okay, so all of us know that this 37 degrees Celsius, as it is a normal human body temperature, at this particular temperature, almost all reaction with a few exceptions are carried out in human body. Okay, so most of the enzyme gives the maximum rate of reaction or its optimum rate of reaction at nearby body temperature. So this temperature coefficient can be studied here, that is Q10. It is said to have a temperature coefficient, and it is a factor by which the rate of biological process or reaction increases for the 10 degrees Celsius increase in the temperature. So for the temperatures over which enzymes are stable, and the rate of most biological processes typically doubles here for a 10 degrees Celsius rise in the temperature. So the Q10 is equal to 2. That is, a 10 degrees centigrade rise in the temperature will increase the activity of the most of the enzyme by 50 to 100% here. So this 50 to 100% increase will be occur here. So which type of curve we get here for such a type of activity? We can see here, that is the effect of temperature over the different molecules. As we know that the reaction rate increases with the temperature to a maximum level, and then suddenly it declines with a further increase in temperature. बघा इथे रेट जो आहे वाढत चाललेला दिसतोय तुम्हाला ग्रॅज्युअली, जसं आपण टेम्परेचर वाढवत चाललो. आणि एक नॉर्मल टेम्परेचर जेव्हा येतं त्याचं ऑप्टिमम टेम्परेचर आपण त्याला म्हणत असतो, की त्या पीकवर, त्या टेम्परेचरवर आपल्याला हायेस्ट पीक जो आहे रेट ऑफ रिएक्शनचा इथं मिळतो. आणि त्यानंतर आपण फर्दर टेम्परेचर वाढवत जरी गेलो तरीही त्याचा रेट जो आहे तो वाढत नाही, तो काय होतो, सडनली तिचा डिक्रीज होऊन जातो. याच्या मागे रिझन आहे. मोस्ट ऑफ द ॲनिमल एन्झाइम्स रॅपिडली बिकम्स deactivated, or we can use the word here denaturation of proteins. As we know that, आपण बघतो की एक साधं उदाहरण आहे, मिल्क प्रोटीन आपण बघत असतो. मिल्क आपण घेतो, त्याला बॉईल करत असतो, आपण गरम करतो आणि मिल्क प्रोटीनला गरम याच्यासाठी केलं जाते की त्यातले जे प्रोटीन्स आहेत ते डिनॅच्युरेशन ऑफ प्रोटीन्स झालं पाहिजे. आणि डिनॅच्युरेशन ऑफ प्रोटीन्स करण्यासाठी अबोव्ह 40 डिग्री सेल्सिअस त्याला आपल्याला वॉर्म करावं लागतं. मग जेव्हा आपण अबोव्ह 40 डिग्री सेल्सिअस त्याला वॉर्म करतो, द डिनॅच्युरेशन ऑफ प्रोटीन, दॅट इज एन्झाइम्स प्रेझेंट इन द milk, that gets denaturate here, and due to this reason we are getting a proper fats from this enzyme or milk here. In similar manner, whatever the enzyme reaction that we are taking here, the optimal temperature of the enzyme in higher organisms which are rarely exceeds the 50 degrees Celsius. Some enzymes loses their activity when frozen, and some enzymes form thermophilic bacteria which are found in the hot spring. तुम्हाला माहिती असतील, एक गरम पाण्याचे झरे असतात, स्पेशली which consisting of sulfur containing moiety and enzyme protein molecules, sulfur containing amino acids त्यात असतात. मग अशा प्रकारचे जे बॅक्टेरिया या हॉट स्प्रिंग वॉटर मध्ये असतात, त्यांची जी ऍक्टिव्हिटी आहे ती आपल्याला 100 डिग्री सेल्सिअस पर्यंत इथे बघायला मिळते. ओके, कारण तिथे काय आहे, त्यांनी सर्व्हाइव्ह केलेलं आहे तिथे आणि त्या पर्टिक्युलर टेम्परेचरला ते तिथे जगत असतात. मग या केसमध्ये आपण इथे बघत असतो की ज्यामध्ये आपल्याला टेम्परेचर जास्त इथे पाहायला मिळतं, त्या केसमध्ये काही एक्सेप्शन जसं असतात ते आपल्याला बघायला मिळतात. आणि जे नॉर्मल एन्झाइम्स आहे आपल्या ह्युमन बॉडीमध्ये, almost all they have the 37 degree as at its particular temperature point having the maximum activity, and after 40 degrees Celsius that activity suddenly goes on decrease or decline graph. So we are getting completely a bell shaped curve. You can see बेल शेप कसा असतो, बेल घंटी असते कशी उलटी लटकवलेली, तशा प्रकारचा कर्व आपल्याला इथे टेम्परेचर मध्ये बघायला मिळत असतो. तर अशा प्रकारचे जे चेंजेस आहे ते रेट ऑफ एन्झाइम कॅटलाइज रिएक्शन्स मध्ये आपल्याला बघायला मिळतात, which is being simply accompanied here as a rise or a fall in body temperature constitutes a prominent survival features for cold-blooded life forms such as lizards or fish whose body temperatures are dictated by the external environment. आपण बघतो की कोल्ड ब्लडेड एनिमल्स मध्ये त्या एक्सटर्नल एन्व्हायरमेंटचा खूप तिथं जास्त इफेक्ट इथं पाहायला मिळतो, स्पेशली लिझार्ड्स मध्ये. लिझार्ड्स या मॅमल्स कॅटेगरी, लिझार्ड्स या कॅटेगरीमध्ये आपण बघतो. इव्हन फिशेस कॅन ऑल्सो बी कम्स अंडर सच टाइप ऑफ कॅटेगरी बिकॉज द इफेक्ट ऑफ वॉटर टेम्परेचर सराउंडिंग देम अफेक्ट और अल्टर्स देअर बॉडी टेम्परेचर अकॉर्डिंगली. एक जनरली आपण बघतो की ह्युमन बॉडीचं सुद्धा तसंच बिहेवियर असते. ओके, ह्युमन बॉडीचं बिहेवियर तुम्हाला थोडक्यात सांगायचं पण आपल्या बॉडीचं जे नॉर्मल टेम्परेचर आहे ते किती असायला पाहिजे, 37. म्हणून आपल्याला बॉडीमध्ये हीट प्रोड्यूस करण्यासाठी मॅक्सिमम फूड तिथं कंझ्युम करावं लागतं. बघा तुम्ही, आपल्या हिवाळ्याच्या सीझन मध्ये म्हणजे विंटर सीझन मध्ये आपला जो डाएट असतो हा खूप जास्त वाढलेला असतो. याच्या मागचं रिझन काय आहे की आपल्या बॉडीचं जे 37 डिग्री सेल्सिअस नॉर्मल टेम्परेचर मेंटेन करायचं असतं, त्यासाठी मॅक्सिमम हीट आपल्या बॉडीमध्ये प्रोड्यूस झाली पाहिजे. आणि प्रोड्यूस होईल आपल्या बॉडीमध्ये जेव्हा आपण प्रॉपर डाएट, प्रॉपर फूड तिथं मिळेल, प्रॉपर तेव्हाच आपल्याला हीट तयार होईल आणि जे सराउंडिंग टेम्परेचरला आपण तिथं हीट ती आहे 37 डिग्री सेल्सिअसला पोहोचू शकतो. याच्याच एक्झॅक्टली अपोजिट आपल्याला कुठं दिसतं, समर सीझन मध्ये. समर सीझन मध्ये आपल्याला भूक जास्त लागत नाही, आपण एकदम लो डाएट तिथं घेत असतो. पण समर सीझन मध्ये बाहेरचं टेम्परेचर जेव्हा 40-45 डिग्री जेव्हा जातं, तेव्हा आपल्या बॉडीच्या नॉर्मल टेम्परेचरच्या एक्झॅक्टली खूप वर तिथं गेलेलं असतं आणि त्यामुळे आपल्याला काय होते, इट अल्टर्स अवर सिस्टीम. म्हणजे आपल्या बॉडीचं टेम्परेचर सडनली वाढायला सुरुवात होते. मग ते आपल्याला तिथं काय करावं लागतं, कमी करावं लागतं. मग कमी करण्यासाठी आपल्याला काय करावं लागेल, इथं पण पाणी प्यावं लागेल. जेव्हा आपण थंड पाणी पितो तेव्हा आपल्याला काय होते, एकदम रिलीफ होतं. तेव्हा उन्हाळ्याचे दिवस असतात, कडाक्याचं ऊन तापत असतं, तेव्हा आपण पाणी पिल्यामुळे आपल्या बॉडीला काय होतं, इथं हायड्रेट करत राहतो कंटिन्युअस. दिस हायड्रेशन विल मेंटेन अवर बॉडी टेम्परेचर. Means both the extreme conditions we have discussed here. In the winter condition, we have to maintain our body temperature at 37 degrees Celsius from the surrounding temperature from 20 to 25 degrees Celsius. In several cases, जसं की आपण हिमालयात वगैरे गेलो तर तिथे मायनस मध्ये टेम्परेचर असतं, इव्हन लेह लडाख मध्ये सुद्धा मायनस टेम्परेचर जातं. मग अशा कंडिशन मध्ये तर ह्युमन सर्व्हाइव्ह नाही झाले पाहिजे असते, कारण आपल्या बॉडीचं जे नॉर्मल टेम्परेचर आहे 37 डिग्री सेल्सिअस असते. तर ते मेंटेन करण्यासाठी तुम्हाला एकदम हाय फ्युएलची इथं गरज पडत असते, हाय एनर्जी फ्युएलची इथं गरज पडत असते. मग जेव्हा आपल्याला हाय फ्युएलची इथं गरज पडणार आहे, इन दॅट केस अवर बॉडी इज ट्राइंग टू मेंटेन द टेम्परेचर टू 37 डिग्री सेल्सिअस. तर हा पॉइंट तुमच्या लक्षात आलेला आहे की हिवाळ्यात आपल्याला भूक का जास्त लागते, बिकॉज अवर बॉडी इज ट्राइंग टू क्रिएट द हीट टू मेंटेन द नॉर्मल बॉडी टेम्परेचर. आणि समर मध्ये आपण पाणी का जास्त पितो, कारण वॉटरचं टेम्परेचर, थंड पाणी जेव्हा आपण पितो ते सडनली काय करतं, सर्क्युलेट होतं आपल्या ब्लड मध्ये इमिजिएटली, आणि ते काय करतं आपल्या बॉडीचं टेम्परेचर तिथं रेग्युलेट करते सो ॲज टू कॅरी आउट द नॉर्मल बॉडी रिॲक्शन. जेव्हा आपल्याला नॉर्मल बॉडी रिॲक्शन्स इथं कॅरी होत असतात, या नॉर्मल बॉडी रिॲक्शन्स जेव्हा डिसऑर्डरनेस त्यात येते, जेव्हा टेम्परेचर फ्लक्च्युएशन होतं, दिस रिॲक्शन इज नॉट बिंग प्रॉपरली कॅरीड आउट, सो देअर इज रँडमनेस to the reaction and disorderness to the reaction. And once the biochemical reactions get disordered, dysfunctioning of the different kinds of biochemical reaction will lead to generate disease condition. म्हणून मग आपल्याला अशा कंडिशन मध्ये प्रॉपरली मेंटेन केला पाहिजे. म्हणून आपला डाएट आणि वॉटर कंटेंट हे प्रॉपरली रेग्युलेट प्रत्येक सीझन मध्ये इथं केल्याच गेलं पाहिजे, असं माझं तुम्हा सगळ्यांना म्हणणं. So this is a very important factor. Again, fourth factor, that is effect of temperature. Now we will move towards the next factor, that is effect of hydrogen ion concentration, that is also known as pH. Okay, pH सगळ्यांनी ऐकलं असेल. Okay, pH स्केल ही माहिती आहे कोणी डिस्कवर केलेली आहे. pH रेंज आपण बघतो pH स्केलवर. काय रेंज आहे, चॅट बॉक्स मध्ये तुम्ही तुमचे आन्सर टाकू शकता. pH स्केलची रेंज काय असते, बिकॉज इट इज टोटली रिलेटेड टू द concentration of hydrogen ion present at a particular time. So what is the range of pH scale? Type your answer in chat box. Very good. Vaishnavi Hudekar, then Sonal Rao, Sakshi Mamankar. Very good. We are counting this scale from 0 to 14. Okay, it is being always said that it is 0 to 14. Vishal Rao, Prem Singh Chavan. Very good answer, beta. आता आपण बघतो की pH स्केल ही 0 to 14 आपण जेव्हा काउंट करत असतो, आपल्या बॉडीमध्ये सुद्धा तुम्ही अॅनाटॉमी आणि फिजिओलॉजी मध्ये एक टॉपिक शिकले असणार, गॅस्ट्रो GIT, गॅस्ट्रो इंटेस्टायनल ट्रॅक्ट. यामध्ये जेव्हा आपण माउथ पासून तर अॅनल्स पर्यंत जेव्हा जात असतो, त्या कोलॉन पर्यंत डिफरंट pH आपल्या बॉडीमध्ये तिथे असतात, आणि डिफरंट pH वर आपलं फूड जे आहे ते तिथे ट्रॅव्हल करत असतं आपल्या पूर्ण बॉडीमध्ये. मग जेव्हा आपण pHचं कॉन्सन्ट्रेशन इथं व्हॅरी करणार आहोत, तर आपल्याला माहिती आहे की त्याचं फूड आपण इंजेस्ट करणार आहे ऍट डिफरंट pH, इट इज हॅव्हिंग द डिफरंट ऍक्टिव्हिटी इन इट. So this different activity can be achieved here on the concentration of pH. So this effect of hydrogen ion concentration, that is the rate of almost all enzymes being catalyzed here, exhibits a significant dependence on the pH value, that is hydrogen ion concentration. And most of the intracellular enzymes exhibits optimal activity or maximum activity at pH values between 5 and 9, and we are getting such a type of bell-shaped curve again as such in the temperature effect. So this effect of pH is also having the exactly inverted shape bell-shaped curve here, and when the activity is being plotted against the pH, a bell-shaped curve is usually obtained here, and this bell-shaped curve we are getting with the different activity of this enzyme. Okay, so as we know that there are different pH in our GIT. So very first we will take here different enzymes are also present. The very first enzyme, whenever food is being ingested by us, it is being kept in our mouth and bitten by the teeth. So we are breaking down this food into the smaller molecule with the help of our teeth, and in the mouth the salivary amylase enzyme is being mixed with that food, and that breaking down the carbohydrates and proteins. So these carbohydrates are broken down in the mouth itself because the amylase enzyme is present in the saliva. Now, what is the pH of saliva? What is the pH of saliva? You can type your answer in chat box. So pH of saliva is around 6.7. Very good, Vishal Rao. It is near to the neutral pH. So as the pH of saliva is very near to neutral, that is 6.7, this amylase is having, the enzyme amylase is having the maximum activity at around pH 7. You can see here the graph. I also taken one another enzyme, pepsin. This pepsin enzyme is secreted here in the gastric juice, especially in our stomach. आणि स्टमक मध्ये जे गॅस्ट्रिक ज्यूस असतात आपले किंवा गॅस्ट्रिक ऍसिड आहे त्याला आपण ऍसिडिक पीएच ला इथं बघतो. म्हणून पेप्सिन नावाचा जो एन्झाईम आहे त्याची ऍक्टिव्हिटी आपल्याला 2 pH ला इथं बघायला मिळते. बघा या कर्व मध्ये तुम्हाला दिसेल, झूम केलंय मी. यामध्ये 2 नंबरचा जो आहे म्हणजे पेप्सिन जो आहे ऍट pH 2 व्हॅल्यू हॅव्हिंग द मॅक्सिमम ऍक्टिव्हिटी पीक. Amylase, the enzyme pH 7, it is having a maximum activity at pH 7, and the trypsin enzyme is having the maximum activity at pH 8.5. There are also certain other enzymes like acid phosphatase. Acid phosphatase is having the activity of pH in the range between 3 to 5. 3 to 5 pH मध्ये आपल्याला acid phosphatase ची activity पाहायला मिळते. In similar manner, alkaline phosphatase is having the activity above pH 9. It is having the activity around 9 to 12 pH value. अल्कलाइन फॉस्फेटेज जो एन्झाईम आहे त्याची ऍक्टिव्हिटी आपल्याला 9 पेक्षा जास्त पुढे गेल्यावर इथं हायर व्हॅल्यूज मिळतात आणि काही अल्कलाइन फॉस्फेटेजची ऍक्टिव्हिटी मॅक्सिमम आपल्याला 9 to 12 च्या मध्ये इथं बघायला मिळत असते. सो these are the factors which affect the enzyme action. And there are also certain exceptions for it, and most of the enzymes, near about 90 to 95% of the enzymes are showing their activity at around the normal body pH, that is 7. नॉर्मल ब्लड पीएच आपण बघतो, ब्लड पीएच सुद्धा आपल्याला अराउंड न्यूट्रल पीएच तिथे बघायला मिळतो, नियर 6.7 to 7.2 या रेंज मध्ये आपल्याला ब्लड पीएच तिथे बघायला मिळतो. मग या पर्टिक्युलर रेंज मध्ये मॅक्सिमम एन्झाईम्स जे आहे ते सुद्धा आपल्याला इथं त्यांची ऍक्टिव्हिटी दाखवताना दिसून येतात. मग यामुळे पीएचचा हा सुद्धा एक इम्पॉर्टंट फॅक्टर जो आहे की जो स्टडी मध्ये आपल्याला इथं आहे आणि ध्यानात ठेवायचं आहे. टेम्परेचर मध्ये जसा बेल शेप कर्व मिळाला, in similar manner the pH or effect of hydrogen ion concentration also shows the bell shaped curve here. So this bell curve in the hydrogen to the reactions of the, so the enzyme proteinase pepsin, proteinase hydrogen ion concentration. Now we are moving towards the next factor, that is effect of activators and coenzymes, or we can also say it is a cofactors effect in it. Activators or modulators in it that we have to study. So the effect of activity of certain enzymes is greatly depending upon the metal ion activators. We can see here there are certain catalysts in organic chemistry. You might have studied certain reactions which are being catalyzed by the metal like nickel, platinum, aluminum. मग आपल्या शरीरात आपण निकेल, प्लॅटिनम, अल्युमिनियम टाकतोय का तिथे? नाही. There are certain other metals in our body like calcium, magnesium, phosphorus. All these are playing a very important role in certain reactions. See for example here, vitamins which acts as a coenzyme in a variety of reactions. There are certain magnesium complex. So this magnesium phosphate which is acting as a coenzyme here. So this magnesium plays a very important role in the ATP. We can say adenosine triphosphate, which is binded to the magnesium, which is attacking towards the glucose here, and this is being showing the reaction by giving its high energy phosphate molecule so as to convert glucose molecule into glucose 6 phosphate. आपल्याला माहिती असेल ग्लायकोलायसिस ची व्हेरी फर्स्ट रिएक्शन जी असते, glucose gets converted into glucose 6 phosphate in the presence of magnesium. The ATP is being consumed here and the phosphatase group is being attached to the glucose molecule. So that very first reaction of this reaction of glycolysis is being catalyzed here by the enzyme alkaline phosphatase here. So this is being carried out at several different modulators or factors in it. So this magnesium plays a very important role in this type of reaction. Apart from magnesium, there are also certain other factors that can also affect this rate of reaction, like calcium. Then magnesium can also alter the ATPase activity and enolase activity in it. There are also certain other metal enzymes in it. We can activate it, like cobalt, then copper, then sodium and potassium ion. You might have seen in certain cases we are going to measure the levels of potassium, sodium, then magnesium by certain pathological tests in it. So this activity of concentration of this metal ion complex can also be altered here. So this is effect of activator or coenzyme. Now effect of modulator or inhibitor. In certain cases, what is happening? This inhibitor competes with the substrate enzyme complex binding sites. In certain cases in our body, there are n number of molecules. These molecules may be similar to that of substrate molecule that is being कन्वर्टेड इनटू प्रोडक्ट. मग हे काय करतात, इथं हे सबस्ट्रेट मॉलिक्युलचा जिथं एन्झाईम साइटवर बाइंड करायचं असते, हे इनिबिटर मॉलिक्युल काय करतात, त्या एन्झाईम साइटवर जाऊन स्वतःला बाइंड करून घेतात. मग स्वतःला जर इनिबिटरने त्या एन्झाईमला बाइंड करून घेतलं तर ऑब्विसली काय होईल, तिथे ऍक्टिव्ह साइट राहील का सबस्ट्रेटला बाइंड करण्यासाठी? मग जर ऍक्टिव्ह साइट राहणार नाही तर सबस्ट्रेट एन्झाईमचा कॉम्प्लेक्सच फॉर्म होणार नाही, आणि एन्झाईम सबस्ट्रेटचा कॉम्प्लेक्सच फॉर्म झाला नाही तर आपल्याला डिझायर्ड प्रॉडक्ट हा तिथे मिळणार नाही. म्हणून जेव्हा आपण बघतो की जेव्हा एन्झाईमला प्रॉपर तिथे सबस्ट्रेट किंवा इनिबिटर बाइंड झाला तर तो रिएक्शन तिथं डिमोलिश करून टाकतो, रिएक्शन तिथेच संपून जाते आणि प्रॉडक्ट आपल्याला मिळत नाही. पण सबस्ट्रेट जेव्हा एन्झाईमला प्रॉपरली बाइंड होतो, इनिबिटर तिथं नसतो, तेव्हा आपल्याला डिझायर्ड प्रॉडक्ट तिथं मिळत असतो. सो ऍज वी इन्क्रीज द कॉन्सन्ट्रेशन और देयर इज अ इन्क्रीज इन द कॉन्सन्ट्रेशन ऑफ इनिबिटर, देन दिस इनिबिशन कॅन अल्टर द रेट ऑफ रिएक्शन. ओके, जसं आपल्याला हे कॉम्पिटिशन चालते सबस्ट्रेट, आपण बघतो की बऱ्याचदा सीईटी घेतल्या जातात, म्हणजे हायर मार्क्सचे मुलं असतात त्यांना चांगली इन्स्टिट्यूशन मिळायसाठी ते चांगले मार्क्स मिळवतात, आणि जे लोअर मार्क्सची मुलं असतात त्यांना तिथं सीट मिळत नाही. मग जिथे काय होते की जेव्हा आपण कॉम्पिटिशन लावतो, कॉम्पिटिशन हा एक असा फॅक्टर आहे की यामध्ये सबस्ट्रेट आणि इनिबिटर सुद्धा इथं कॉम्पिट करत असतात फॉर गेटिंग द प्रॉपर साईट लाईक एन्झाईम. मी तुम्हाला आधीच सांगितलं की एन्झाईम इज ऍक्टिंग ऍज अ प्रॉपर टीचर, आणि काही टीचरच्या ठराविक बॅचेस असतात की जसं की आपण म्हणतो की मला हा चांगला टीचर भेटला पाहिजे, मग सबस्ट्रेट असतात आणि सबस्ट्रेटच्या सोबत असंख्य अनेक मॉलिक्यूल सुद्धा असतात. मग बाकीचे मॉलिक्यूल जर त्या एन्झाईम बॅच मध्ये जर अकोमोडेट झाले तर सबस्ट्रेटला तो प्रॉपर टीचर भेटणार नाही, आणि त्या स्टुडंटला प्रॉपर टीचर भेटला नाही तर प्रॉपर आउटपुट आपल्याला मिळणार का? नाही. मग प्रॉपर रिझल्ट तिथं मिळणार नाही. म्हणून प्रॉपर स्टुडंटला एक ठराविक इन्स्टिट्यूशन तुम्हाला मिळालं पाहिजे, जसं की आपलं इन्स्टिट्यूशन तुम्हाला इथं मिळालेलं आहे, जेणेकरून तुमचं लाईफ तिथे चांगलं बनेल. मग इथे आपल्याला सबस्ट्रेट एन्झाईम कॉम्प्लेक्स जसा फॉर्म होईल त्याच प्रकारे इथे आपल्याला इनिबिटरचा आणि एन्झाईमचा कॉम्प्लेक्स फॉर्म होतो, पण त्याच्यापासून आउटपुट मिळत नाही त्या मुलाला प्रॉपरली तिथं, किंवा त्या एन्झाईम इनिबिटर कॉम्प्लेक्सला प्रॉपर प्रॉडक्ट तिथं मिळत नाही. मग त्यामुळे जी रिएक्शन आहे ती तिथं इनिबिट केली जाते. या एन्झाईम इनिबिशनचा सुद्धा एक स्पेशल टॉपिक तुमच्या सिलेबस मध्ये आहे, मेकॅनिझम ऑफ एन्झाईम इनिबिशन. त्यामध्ये सुद्धा 3 टॉपिक्स असतात, कॉम्पिटिटिव्ह इनिबिशन, नॉन कॉम्पिटिटिव्ह इनिबिशन अँड थर्ड वन इज ॲलोस्टेरिक इनिबिशन. तर अशा प्रकारचे रिएक्शन एन्झाईम जी आहे ऍक्टिव्हिटी ती आपल्याला स्टॉप करता येतं. तर ड्यू टू टाइम कन्स्ट्रेंट आय एम नॉट गोइंग टुवर्ड that point. So we are moving towards our last point, that is factors affecting enzyme action, that is effect of time. The longer as the enzyme time, we can see here, as the longer as the enzyme is being incubated with the substrate, the greater the amount of product that will be formed here. So here the rate of formation of product is not a simple linear with a function of time of incubation. जसं आपण जास्त वेळ दिला रिएक्शनला तर आपल्याला डिझायर्ड प्रॉडक्ट मिळेलच याची खात्री नाही, ठराविक वेळ जेवढी लागते तेवढी तिथे देणं गरजेचं असतं. पण जेव्हा एक्सेस टाइम तिथे लिहिला, जसं तुम्हाला पेपर जो लिहायचा आहे, एक्झाम मध्ये तुम्ही पेपर सॉल्व्ह करता, तुम्हाला लिमिट राहते 3 आवर्स मध्ये तुम्हाला कम्प्लीट पेपर तुमचा सॉल्व्ह करायचा आहे. पण 3 आवर्स पेक्षा तुम्हाला जास्त टाइम दिला तर तो काही फायद्याचा राहतो का? तुम्हाला 3च तासात तुमचं तिथे जे आहे ते द्यावं लागतं. मग त्यामध्ये जास्त टाइम देऊन त्याच्यात जास्त फायदा होत नसतो, त्यामुळे तिथे तुमचं प्रोडक्टिव्हिटी जे आहे ते लॉस होत असते. सो दिस एन्झाइम कॅटलाइज रिएक्शन्स अंडरगोज द कम्प्लिशन इन ऑप्टिमम टाइम and variation of time does affect the rates of enzyme catalyzed reaction according to this graph. You can see a time is being given here and enzyme catalyzed reaction are smoothly carried out. And if we give more time and lot of time, it is being not giving a proper product. It is achieving again the steady state concentration of the product in it, because it is altering the several factors in it. So this is all about the different factors that we have studied here. So we will take a recall here and which points that we are going to take home from today's lecture. So what we have studied, that is different factors which are affecting the enzyme concentration. Very first factor is concentration of substrate, which is initially directly proportional, and later on it is following the zero order reaction, following the same manner for the concentration of enzyme. Then concentration of product, it is also being affecting the rate of reaction. Then temperature, at normal body temperature, most of the enzyme gives us a maximum reactions in it. Then pH is also having the bell-shaped curve. Then time, and finally we are discussed about the presence of activators or inhibitors like the coenzymes or modulators in it. So this is all about my today's talk, that is factors affecting enzyme activity. So if you have any queries, please let me know. You can also type it in chat box, or you can unmute yourself. If you do not understand any point, you can ask to me regarding this concept. So thank you very much for attending this today's lecture very calmly and nicely. If you have any doubt, please let me know. I'm waiting for your questions. Thank you very much. You can type your questions in chat box, or you can directly unmute yourself and ask me any doubt. Hope so. I think all of you understood this concept. This is a very, very important topic. If you have to write a question and examination, this is a compulsory question initially. प्रत्येक एक्झाम मध्ये तुम्हाला फॅक्टर्स अफेक्टिंग एन्झाइम ऍक्टिव्हिटी 10 मार्कासाठी विचारला जायचा. आता हे एमसीक्यू बेस्ड क्वेश्चन्स आलेले आहे म्हणून यात तो विचारला नाही जात. काही फॅक्टर्स मध्ये तिथे तुम्हाला विचारला जाऊ शकतो. तर या केसमध्ये तुमच्या फर्दर स्टडीज साठी हा खूप महत्वाचा आहे, म्हणून बायोकेमिस्ट्री हा खूप इम्पॉर्टंट सब्जेक्ट तुम्हाला इथे शिकायचा आहे. Because biochemistry tells us about the life cycles which are, or whatever the chemical reactions which are occurring inside the human bodies. So that's why So if you don't have any doubt, I'm thanking very much to the organizer, especially Cheke sir, Ambore madam, those who have given me opportunity to deliver the talk over this topic. And finally, also thanks to the honorable principal sir, so as to give such a type of wonderful opportunity for other teachers to interact with the different students, because we are taking lectures over the other students, and this is a very important learning from the other teachers too. So for you peoples, this is a beneficial, I think. Okay, so if you have any doubt, please feel free to ask. Raise your hands, unmute yourself later on and ask me, or otherwise we will stop here. Over to you, Cheki sir, Ambore madam.
Thank you very much, sir, for giving us such a wonderful information. Now, if anyone have any question, please raise your question in chat box. If anyone.
बघा, तुम्हाला काहीही डाउट्स असतील तर बिंदास विचारा. आपल्याला डाउट्स असतील, ते डाउट्स आपले क्लिअर झाले तर ते लाईफ लॉंग आपल्या लक्षात राहत असतात. त्यामुळे जर समजलं असेल माझं शिकवलं तर मग काही अडचण नाहीये. पण तुम्हाला काही याच्यातही डाऊट्स असते तर बिंदास तुम्ही तिथे विचारा, जेणेकरून तुम्हाला इथे फर्दर फ्युचर मध्ये चांगला इथे स्टडी मध्ये हेल्प होईल. कारण सर्व बायोकेमिकल रिॲक्शन्स एन्झाईम ऍक्शन वरच डिपेंड करतात. ते चॅप्टरच तुमचा पूर्ण या बायोकेमिस्ट्री मध्ये इम्पॉर्टंट चॅप्टर म्हणून कन्सिडर केल्या जातात. ओके, सो इफ एनीवन हॅव्हिंग द डाऊट प्लीज फील फ्री टू आस्क.
I think so there is no question. So
Yeah.
I would conclude this session. It give the image.
Just a minute, ma'am. Just a
To
minute.
deliver the vote of thanks.
Just a minute, madam. There is one question in a chat box asked by the Sakshi Mamankar. Activation energy सांगा sir. Okay. The activation energy एक आपण स्लाइड घेतलेली आहे का याच्यात मला बघूया. ओके, इथे बघा, कुठलीही केमिकल रिॲक्शन जेव्हा होत असते या केमिकल रिॲक्शनला कॅरी करण्यासाठी, म्हणजे एक जो आपण स्टार्टिंग प्रॉडक्ट म्हणतो त्याला आपण रिस्टार्टिंग सबस्टन्सेस जे म्हणतो त्याला आपण रिॲक्टंट म्हणत असतो. मग रिॲक्टंटला प्रॉडक्ट मध्ये कन्वर्ट करण्यासाठी आपल्याला सर्टन अमाऊंट ऑफ एनर्जी सप्लाय करावी लागतं. से फॉर एक्झाम्पल एखादं तुम्ही सिंथेसिस करत आहे ज्यामध्ये तुम्हाला हीट द्यावी लागते, आणि ही हीट जेव्हा देतो असतो आपण, ही हीट देत असताना हीट पासून ती एनर्जी त्यांना मिळते आणि ते मॉलिक्युल्सचं ऍक्टिव्हेशन तिथं होत असतं. जनरली मॉलिक्युल्सचं ऍक्टिव्हेशन ज्या पॉइंटला होतं बाय सप्लाइंग सर्टन अमाऊंट ऑफ एन्थाल्पी, दॅट हीट इज नोन ऍज enthalpy, and that is point is known as activation energy. And once the substance is being activated, it is converted into the transition state. This transition state is one of the intermediate point between the substrate and product. It is not completely a substrate, not completely a product. म्हणून त्याला इंटरमिजिएट फेज म्हटलं जातं. आणि त्या इंटरमिजिएट फेजला जेव्हा ते ऍक्टिव्हेशन एनर्जी आपण प्रोव्हाइड करतो, सर्टन अमाऊंट ऑफ एनर्जी त्याला प्रोव्हाइड केलं जाते, देन अँड देन ओनली इट इज सेड टू हॅव अ ऍक्टिव्हेशन एनर्जी. अ व्हेरी गुड क्वेश्चन बेटा. दिस इज अ व्हेरी गुड क्वेश्चन आस्क्ड बाय यू. ऑर्गॅनिक केमिस्ट्री हे कुठे शिकलं जातं आपल्याला? ऑर्गॅनिक केमिस्ट्री मध्ये इथं आपण हे शिकत असतो. आणि ऑर्गॅनिक केमिस्ट्री मध्ये हे जे आहे ऍक्टिव्हेशन एनर्जीज आपण बघत असतो. तुम्ही SN1 आणि SN2 रिएक्शन शिकले असणार, त्यामध्ये आपण बघतो की रेट ऑफ रिएक्शन जे असतात ते डिपेंड करतात डिफरंट इंटरमिजिएट्स फॉर्मेशन मध्ये. आणि SN2 रिएक्शन इट इज अ सिंगल स्टेप रिएक्शन, आणि त्यामध्ये जसा एक ब्रिज इंटरमिजिएट फॉर्म होतो. सो दॅट इज हॅव्हिंग अ मॅक्सिमम एनर्जी. इंटरमिजिएट जे असतात हायली अनस्टेबल असतात. हायली अनस्टेबल आहे म्हणजे त्यांची एनर्जी ही सर्वात जास्त आहे, आणि त्यानंतर ते प्रॉडक्ट मध्ये कन्वर्ट होतात आणि त्यांची जी एनर्जी आहे ती कमी झालेली आपल्याला इथं दिसून येते. सो इफ वी हॅव द लोअर ऍक्टिव्हेशन एनर्जी, दॅट मीन्स मोर मॉलिक्युल्स हॅव द रिक्वायर्ड एनर्जी टू रीच द ट्रान्झिशन स्टेट. म्हणून ऍक्टिव्हेशन एनर्जी ही कधी पण आपल्याला इथं जास्त असली पाहिजे. आणि एन्झाईम मुळे आपण इथे बघितलं या ते बघा, ऍक्टिव्हेशन एनर्जी फार कमी लागते जेव्हा एन्झाईम प्रेझेंट असतो, आणि एन्झाईम अब्सेंट असला वरच्या फिगर मध्ये ए मध्ये आपल्याला ऍक्टिव्हेशन एनर्जी खूप जास्त लागते. म्हणजे खूप अमाऊंट ऑफ हीट किंवा तिथे आपल्याला फ्युएल सप्लाय करावं लागेल. साधं एक उदाहरण घ्या, तुम्ही सरळ रोडवर तुम्हाला गाडी चालवायची आहे तर तुम्ही नॉर्मल टॉप गिअरला ती तिथे चालवता. पण तेच म्हटलं तुम्हाला एक इन्क्लाइन पोझिशन घाट लागला आणि त्या घाटाला वर चढायचं आहे, तर आपल्याला तिथं काय करावं लागतं? गिअर शिफ्ट करावं लागतं लोअर गिअरवर. आणि लोअर गिअरवर जेव्हा आपण शिफ्ट करतो तेव्हा आपल्याला तिथं जास्त सप्लाय ऑफ एनर्जीचं द्यावं लागते. आणि प्लेन 1 किलोमीटर चालणं आणि 1 किलोमीटरचा घाट चढणं यामध्ये खूप फरक तिथं आपल्याला मिळतो. त्याला जास्त फ्युएल लागतं प्लेन रोड पेक्षा वर चढण्यासाठी. इन सिमिलर मॅनर ऍक्टिव्हेशन एनर्जी जेव्हा जास्त लागणार तेव्हा आपल्याला इथं रोल जे आहे कमी पाहायला मिळत असतात. तर समजलं मग सगळ्यांना इथे हे ऍक्टिव्हेशन एनर्जीचा कन्सेप्ट? साक्षी क्लिअर झाला असेल तर पुन्हा विचारा, झालं असेल तर येस सर म्हणा. ओके म्हटलंय तो. ओके, व्हेरी गुड बेटा. अजून कोणाला काही डाऊट असतील तर विचारू शकता. That's why I taken this diagrammatic representation for you people so as to understand the concept of enzyme substrate complex formation easily. Over to you, madam.
Okay. Thank you very much to one and all. It gives immense pleasure to deliver a vote of thanks of this session. I would like to thank chief guest or our speaker, Dr. Vaibhav Adhao sir, for enlightening us such a wonderful knowledge with along with the proper presentation on a topic factor affecting the rate of reaction. I would also thank to dear participant, dear student for listening carefully. And also, I would like to thank our beloved principal sir and our organization for arranging a such session for our student. Once again, I thanks to all dear student, and once again, I also thank to Dr. Vaibhav Adhao sir. Thank you so much. With the permission of Dr. Vaibhav Adhao sir, I conclude this session. Thank you.
Yeah, thank you very much, one and all, to listen it carefully. Even though in future, if you have any doubt, feel free to call me. Okay, thank you.
Thank you.